In this section, we're talking about the structure of chromosomes. So there are several terms that you need to make sure you understand in order for any of this to make sense. First of all, chromatin is the form that DNA is found in before it's replicated. So this is when it's not in a chromosome form, it's just kind of a jumble, unwound like a ball of yarn that's kind of in a wad. So a chromosome is once the DNA has gotten wound up, it's all wound up tight and condensed into a chromosome form. And this only happens right before mitosis. So the DNA is actually wound around several proteins that are called histones. And each group of histones with the DNA that's wound around it is called a nucleosome. So let's have a look at a bigger picture of that. So this is a chromosome when it's all wound up together around the histones. Each of these is a histone. And so each nucleosome is like here and here, and so it has several histones and then the DNA that's wound around with it. So each of these bits here is called a nucleosome. So sister chromatids are copies of each other, and they're made before mitosis. So these are sections of chromosomes that are duplicated. So they're identical copies of each other. And the centromere is a little bunch of stuff that holds the sister chromatids together. So in this picture you can see each of these here is a sister chromatid, and this here in the center that's holding them together is the centromere. Homologous chromosomes have genes that code for the same characteristics in the same locations. So, for instance, you're looking at 8 pairs of homologous chromosomes here, and let's say there was a gene for eye color right here. This is called the locus. This is the location on each chromosome. And so what the homologous chromosomes have are identical copies of the same gene on the same place for each homologous chromosome. So each sister chromatid has a copy of the same thing in the same place. They're about the same size, about the same shape, and they pair up right before mitosis happens. So on homologous chromosomes, there are what we call alleles, and these are actually variations of a gene, and they're found each homologous chromosome has one for one particular gene, and they're found in the same place on each gene. So one example would be a gene that codes for hair color, and there might be an allele for black hair and an allele for blonde hair. So ploidy is another term you need to understand, and ploidy is showing the number of copies of a gene or chromosome in an organism, and we abbreviate this with a number and the letter N. So a haploid individual, we say this is 1N, and this individual has one copy of each gene. A diploid individual, we say is 2N and has 2 copies of each gene. So in this picture here, for instance, this is a 2N individual because, as you can see, there are 2 copies of this green chromosome, which each have the same gene on them, and 2 copies of this yellowish chromosome, and each of those have the same gene on them. So this individual is 2N. So from the moment of fertilization, humans are 2N. Now that means that each of our sperm and eggs are haploid. So haploid is 1N. So if this was, let's say, an egg cell, it's 1N because each one of these is only one copy of each chromosome. So there's one there's one here, and there's one over here. And so finally we talk about the human karyotype. And a karyotype is a picture of chromosomes. For humans, the 2N number is 46. So that means we have 23 chromosomes. Number 23 are our sex chromosomes, which in this picture are here. And you might notice that the X chromosome, which is this one, is larger than the Y chromosome, and that's normal in people. But if you look at the other chromosomes, you'll see that the majority of them are roughly the same size and the same shape. So make sure you have a look at the complementary reading material for more information about karyotypes and chromosome folding and ploidy.